The next chapter, Chapter 7, in our pursuit of pathologic harmony, is immunopathology. And so what are the things that they love to ask on the USMLE that are critical to your understanding, which is also going to be discussed in immunology, are those hypersensitivity reactions. And as mentioned previously, the hypersensitivity reactions become critical in understanding the pathogenesis of a number of very important diseases that you will see reiterated and revisited multiple times through the organ systems. So the first hypersensitivity reaction, type 1, goes by various names, immediate or anaphylactic type. This is very easy to remember because it involves what unique antibody on the surface of mast cells is IgE, and basically how many Ig molecules that you can find on one single mast cell, up to 10,000 can be actually attached to one mast cell, so it wouldn't take much of a trigger to release all the chemical mediators of inflammation once the antigen is bound to the IgE on the surface of the coated mast cell. Obviously, this requires previous sensitization to the antigen, so the history and the clinical vignette would illustrate this process. And then eventually you get basically requires cross-linking of the IgE Fc receptors on the surface of mast cells and basophils to allow it to release the various constituents within the granules of the mast cells. And when you release these chemical mediators, what are these mediators? This is a review of what we talked about in acute inflammation. Obviously, a very potent vasodilator would be histamine, and heparin is also found in mast cells, an anticoagulant. Then it also will attract inflammatory cells, so you have eosinophilic chemotactic factor. You will also release leukotrienes, including leukotriene B4, remember, is important for chemotaxis, as well as upregulating adhesion molecules on leukocytes. And you'll also release another chemotactic factor for neutrophils as well. Then the prostaglandins like D4, platelet activating factor, and leukotriene C4 and D4 will also be released as a consequence of this degranulation from the mast cells. And then as more and more eosinophils are recruited in, obviously you're going to also increase vascular permeability, so the area will become swollen or you'll get edema. And so this would be an exudative type of issue that one would see because of eosinophils and neutrophils and a high protein content. And it can either be localized, like atophy, skin, or, for example, systemic anaphylaxis that could obviously dire consequences leading to massive increased vascular permeability, leading to anaphylactic shock. And it can be things like antigens, previous bee stings, drugs, are examples that could lead to this type of immediate response. What's immediate? This can happen within seconds to minutes. So that's the time frame you have to think of in which one would see this hypersensitivity reaction. For example, a bee sting, the swelling can occur within minutes after the sting for those that are sensitized to the bee sting itself. And then we talk about food allergies or atophy, and then a classic example in the respiratory tract is asthma or reversible reactive airway disease, which we'll talk further about in the respiratory tract section. Now we come to hypersensitivity reaction type 2 or antibody mediated. Now this becomes important because 2 and 3 are the ones that usually confuse the students, especially recognizing the clinical vignette what's the underlying mechanism. So they can be subtyped, especially type 2 or antibody mediated, but the general rule is this. Type 2 should be thought of as an antibody bound to a target cell or tissue, because what can be activated in the Fc portion of the antibody, as you know from immunology, is going to be complement, and then you can lead to acute immunologic damage. And what type of necrosis do you associate with this would be fibrinoid necrosis that we discuss in the second chapter of pathology. So basically, the antibody can be either IgG or IgM to a specific target cell or tissue, and that's the critical definition that you must always realize, and then be able to recognize it, obviously, in the clinical setting to be able to answer the question correctly. So it's complement-dependent cytotoxicity, as I mentioned before. You get activation of complement on the Fc portion. especially when you have now allowed the antibodies to coat the surface of cells, like red blood cells, for example, where you can get hemolysis of the red blood cell, and there's antibody-dependent cell-mediated cytotoxicity, where you get cytotoxic killing of antibody-coated cells. Another subtype of hypersensory reaction type 2 is anti-receptor antibodies, where the antibodies can either activate or interfere with the receptors. What are classic clinical conditions that you learned about that are illustrative of this would be antibodies to acetylcholine receptors in the motor end plates that leads to myasthenia gravis, for example. Or you think of Graves' disease, where you have antibodies to the TSH receptor leading to hyperstimulation of the follicular cells in the thyroid and leading to hyperthyroidism. Type 3 hypersensitivity is immunocomplex disease. And how do you differentiate this with hypersensory reaction type 2? Remember, the easy way to remember this is very simple, is that here we have an antibody bound to a soluble molecule. And now it can either form in tissue, and immunocomplexes, since we have two bound molecules, can now, as a complex, circulate and get deposited anywhere in the body. There's a propensity for these immunocomplexes to now be deposited and lead to damage by the FC portion activating complement and then producing fibrinoid necrosis is a typical example of damage to the tissue. When we talk about hypersensory reaction type 4, we think of C-cell mediated type. There's two subtypes that are important. We've talked about a number of these. The one you always think about, delayed type hypersensitivity, it takes time for this process to take place because it's recruiting macrophages, and this involves granuloma formation that we talked earlier, and chronic inflammation, where it requires CD4H1 helper cells to release gamma interferon that's going to recruit the monocyte from the intravascular space to become activated macrophages, or what we call epithelioid cells. So you think of conditions like tuberculosis, coccidioidomycosis, leishmaniasis, leprosy, as well as the gammas found in tertiary syphilis. And what MHC class does H1 or CD4 positive cells interact? Remember, it's going to be MHC class 2. And we think of examples like PPD skin test, and for TB, cocci, or tuberculosis itself, or other conditions that I just mentioned. The second subtype of hypersensory reaction type 4 is what we call the cytotoxic T-cell mediated. This obviously involves cytotoxic T8 cells that basically interact with what MHC class is going to be class 1. What's the one cell in the body that does not have MHC class on it is red blood cells. So when we think of cytotoxic T8 cells, it's going to obviously cause lysis or destruction to those cells that have some type of antigen that's on the surface of the cell, complexing with the MHC class 1. And what do we think of? Viral infections. We think of the tumor-associated antigens on the surface of malignant tumors that can cause an inflammatory response. Contact dermatitis, where you're sensitized to some type of toxin or molecule that's going to have the cytotoxic T8 cells in the skin react. Graft versus host disease, when you have bone marrow transplantation where the cytotoxic T8 cells from the donor bone marrow will now destroy the recipient cells like the keratinocytes in the skin or the enterocytes in the small intestine like the duodenum, as well as hepatocytes and bile duct cells that you can see in the liver.